Welcome back to CHM5403 Sensor Technology. Today we will discuss immobilization strategy for biomarker or chemical marker. Immobilization strategy of biomarker is very important because it will definitely affect the response or the signal generated by the biomarker. In considering the strategy for immobilization, we must consider a few things, which are the substrate or what kind of material involved as the transducer, the method of immobilization, which chemistry, the condition, the pH or supporting electrolytes or the buffers used in the experiment, as well as the biomarker itself. For example, if we are using DNA biomarker, then the length of the probe, the packing, and the linker must be considered, must be characterized. In order to have an optimum signal for biosensor, the restriction of the biomarker or chemical marker on top of the transducer must be reduced. In other words, the immobilization, the amount of DNA, antibody, aptamer, or chemical marker must be optimized. It does not have to be too many as well as too little. And the immobilization must be stable, means it does not have the capacity to detach from the sensor surface after washing process. And when we immobilize the biomarker or the chemical marker, we need to ensure that the biomarker or the chemical marker is accessible to the target of interest. And the last point is we have to consider the non-specific binding or interference from non-interest analyte. These are some of the strategies that we can use to immobilize the biomarker or the chemical marker on top of the transducer or sensor surface. One is simple absorption. Second one, we can use polymer. The third one, we can use the interaction of biotin, avidin, tetravidin, the use of nanoparticle, as well as self-assembled monolayer. Simple absorption, especially usually used if the sensor surface is carbon-based. It is used because it is simple, but the problem with simple absorption is the orientation of the biomarker. Since DNA requires an endpoint attachment of the DNA probe, for example, then the simple absorption is not practical for DNA biosensor. As well as immunobiosensor, it requires the antibody is attached to certain specific orientation on top of the sensor surface. Then again, simple absorption is not practical for those two types of biosensor. This is what happens if you use simple absorption for DNA-based biosensor, where the DNA probe will not be standing up, or the DNA probe will not have an endpoint attachment like the right side figure on top of the sensor surface. The endpoint attachment of the biomarker, in this case DNA probe, is very important to ensure the interaction of this DNA probe with the target. So what happens if we choose simple absorption, then the DNA probe will be probably lying down like the left side figure. These are the problems with simple adsorption. Hybridization efficiencies probably very low. That will affect the detection limits. And definitely the DNA probe will be pinned down instead of endpoint attachment. What about polymers? Using polymer to immobilize the DNA antibody biomarker. So by principle, the biomarker as well as the chemical marker can be entrapped within a polymer structure. Polymer has certain porosity that gives the exposure of the biomarker or chemical marker towards the target analyte. However, due to the size of the porosity, it will sort of inhibit the target analytes or target DNA to be able to diffuse into the polymer to interact with the DNA probe or biomarker inside the polymer. So imagine that this is the DNA probe that is immobilized inside the polymer porosity. And imagine that the target DNA has to diffuse inside here to be able to hybridize with the DNA probe. So the resistance will inhibit the process of hybridization for DNA-based biosensor. Another strategy is by using positively charged polymers. For example, we have chitosan, polyaniline, polypyrrole, polythiophene, as well as other conjugated polymers. Since the charges of the polymers are positive and the charges of the DNA probe, the phosphate backbone is negative, then there is electrostatic interaction between the positive charge of the polymer with the negative charge of the phosphate backbone of the DNA. This can also be utilized as a mean of immobilization, but however, the issue here again is it does not enable endpoint attachment of the DNA as well as biomarker like antibody. So these are the disadvantages of using polymers as the mean to immobilize the biomarker, where due to the certain sizes of the porosity, it is possible that the target DNA or the target analyte cannot diffuse into the polymer membrane or the porosity of the membrane of the polymer. This will prevent the hybridization, if it is DNA, to occur or interaction with the biomarker to occur. Hence, this will affect the signal of the biosensor. Covalent attachment is a very good alternative for immobilization of DNA, antibody, as well as chemical marker. As you can see here, we have carbon surface, and that carbon surface is modified with certain chemicals, which results in the carboxyl group in the end of the electrode. And let's say we have aminated DNA, means that the DNA probe has aminated N, okay, and with the help of EDC NHS, then an amide bond can be formed between the carboxyl group on top of the carbon electrode with the amine end of the DNA probe. This will ensure a very stable immobilization on top of the sensor surface, as well as an endpoint attachment of the DNA, in this case is a DNA, on top of the sensor surface. These are the example of endpoint modification of the DNA probe that we can order straight from the companies. As you can see here, we have thylated N DNA probe, aminated N DNA probe. So before ordering the DNA probe, you have to consider what kind of chemistry involved on the surface of the sensor. Let's say we have carbon nanotube, which is carbon, okay? And this carbon nanotube can be treated with strong acid, where carboxyl functional group will be formed on top of the carbon nanotube surface after treatment of acid. And with activation of EDC NHS, okay, the carboxyl group can form amide bond between the carboxyl as well as with the amine from attached to the protein or attached to the DNA from the biomarker. So carboxyl with activation of EDC NHS will form an amide bond with the amine group from the protein or DNA. Another strategy is by using gold surface, where we can have self-assembled monolayer by utilizing thylated N of the biomarker. So in this case, we can order thylated DNA, so that we can easily do the self-assembled monolayer process on top of the gold surface. It is known that thiol has a very good affinity towards gold surface, where it can form pseudo-covalent bond. If you can see here, the red dots is representing the thiol groups here. So by right, if you expose the thylated DNA with gold surface, they will automatically self-assemble on top of the gold surface. But unfortunately, there are a few issues because you might end up having the DNA or biomarker to behave like this. So in order to avoid this kind of behavior, this kind of problem, we need to have substrate passivation. The substrate passivation can be done by treating the goal surface with a negative charge compound. For example, in here we use alkane thiol. So the thiol group will be attached to the surface of the gold, and we have the alkane group on top of the gold surface, right? And this will serve as passivation to avoid the lying down or the pin down of DNA or any biomarker, as well as it will serve as to repel unwanted species or non-specific binding to the surface of this sensor or goal. This is the protocol that you can use to do the self-assemble monolayer process. First, by cleaning the gold electrodes and incubating the gold electrode in thylated DNA probe. Further incubation with alkane thiol as the surface or substrate passivation, followed by rinsing or washing. Let's say you wish to use aminated DNA instead of thylated DNA, and you wish to use self-assembled monolayer. So certain activation or certain modification, chemical modification, has to be done on the surface of the gold. So in this case, these are suggested protocol that you can use to modify the surface of the gold, whether it is electrode at any gold surface sensor. In this case, we can use mercapto and decanoic acid to provide the carboxyl functional group on top of the gold surface, and followed by activation with EDC NHS, where we can have an amide group form between the carboxyl group with the amine from aminated DNA. This is an example of using aminated DNA. So certain functional group has to be modified on the surface of the gold, and with that groups can be reacted, forming an amide group with amine from the aminated DNA.